So welcome to Chapter 6. Chapter 6, we're going to be talking all about protein, and we're going to begin with protein structure. Now, in order to talk about proteins, we need to first talk about amino acids, because those are the building blocks of all of our proteins. We need to understand all about an amino acid to kind of understand a little bit more about how they work in terms of building proteins. So let's start with our generic amino acid. And if we take a look to its left here, we're going to see that it contains an amino group. And the amino group is interesting because it contains hydrogen, but it also contains nitrogen. And we're going to see that nitrogen is the element that sets proteins apart from our carbohydrates and our lipids. Hold on to that thought, kind of tuck that away, because we'll come back and talk about a nitrogen balance as one of the ways that we might be able to assess our protein status. So that's the amino group. Then on the other side, we have an acid or a carboxyl group. And that acid or carboxyl group is made up of carbons, oxygens, and hydrogens. So when we look at all of our amino acids, we're going to see that the amino and the acid groups are going to be the same on all of our amino acids. However, they're also going to contain a carbon skeleton, and that carbon skeleton is going to be different for each of our amino acids, and that's what's going to make our amino acid unique. If we take a look at our two examples, glycine and L-alanine, we'll see exactly that our glycine, for its one little piece on the carbon skeleton, contains a hydrogen, while L-alanine contains carbon and 3 hydrogens in its carbon skeleton. Now, it's also important to understand how the amino and the acid groups interact. We will find that they are actually attracted to each other. And so when we're putting together an amino acid to create a protein, we're going to want that to happen. So our amino acids are going to be lined up amino to acid to amino to acid, etc., so that we have those attractions, and then that's where our chemical bonds are going to form. Now, as I've mentioned, our amino acids are our building blocks of proteins, and it's important to understand the fact that there are 20 different amino acids that make up all proteins. And I always think of this kind of like the letters of the alphabet, because we know we have only 26 letters in our English alphabet, and yet we end up with literally thousands of different words based off of the combinations that we put those 26 letters into. So the same thing is absolutely true for our amino acids and our proteins, that we can take those 20 different amino acids and we can make literally thousands of different proteins that have different structures and hence different functions. 9 of those 20 are what we call essential amino acids. An essential amino acid is something that your body cannot synthesize, and so it must be eaten in our diets. If you recall back to fats, we talked about essential fatty acids as well. So that means that the other 11 are non-essential amino acids, and they are synthesized in our bodies, assuming, of course, that we have the right raw materials in order to make that happen. On average, our proteins are going to supply us with 4 calories per gram in terms of energy. So as we are breaking those chemical bonds, they're peptide bonds, if you are interested in what they're called, those bonds that are going to hold our amino acids together, as we break those, we're going to be getting energy in the amount of 4 calories per gram. So very similar to what we got from our carbohydrates. Now let's come back and talk a little bit more about those essential amino acids. And again, there are 9 of them, and they are things that we can't synthesize by our body in sufficient amounts or at all. Some of them we can do just a little bit, but not enough to really meet our needs, and so we rely on our diet. Now, many times we talk about a limiting amino acid, And this is the essential amino acid that is in the lowest concentrations in the food or our diet relative to our needs. So when we are looking at other things like perhaps making a new protein, this is going to be the one that will limit the amount that we're able to make if we don't get enough in our diet. Now, sometimes amino acids are what we call conditionally essential, which means most of the time we can make them. However, if we don't get enough in our diet of the raw materials, then we're not going to have the right amounts to be able to make that. So conditionally essential are really those amino acids that are made from other essential amino acids if insufficient amounts are eaten. So classification of amino acids, just in case you ever wanted to know and amaze your friends at parties, we can talk about those essential versus non-essential. And certainly for this course, you're not going to be required to memorize those. You'll notice that some of those essential amino acids have a little asterisk next to them that says they're a branched chain amino acid. And hold on to that thought because we're going to come back and talk about those in a second. So our essential amino acids are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Our non-essential amino acids are alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. Now, you may have heard of a few of those for different reasons, maybe in food products, others reports on things. I think tryptophan is one that gets a lot of press like around Thanksgiving time because it's in high concentrations of turkey and other protein foods. But those are the essential versus non-essential amino acids. Now let's talk a little bit more about those branched chain amino acids. And these have a slightly different chemical structure. So if we think back to that carbon backbone that we looked at, that was the piece in the center that had the unique characteristics or the unique structure for each individual amino acid. We're going to see that these have a little bit more of a branch to them. They're not necessarily that much different, but that individual piece is going to have a little bit bigger structure and a little bit more to it. So that is going to influence how our body is going to use them. Now, remember from our previous slide with the little asterisks, we had leucine, isoleucine, and valine were our branched chain amino acids. And not only are they essential, but they are used by our muscles for energy. So we're going to find that they are very popular with strength training athletes because the thought is that the more branched chain amino acids I take in, not only does that help me with my muscle building, but it also may help me with more energy while I'm working out and certainly helps with muscle recovery when I'm done. So it is found, the branched chain amino acids are found in many things, but they're found in high concentrations in the whey protein from milk, again, making it a popular supplement with a lot of athletes. We'll also find that branched chain amino acids are used in the medical setting many times for that critically ill patient as a way of helping them with their recovery from their injury or their illness. Next, let's talk about how we're going to put all those amino acids together and organize our proteins. So the order in which we put those amino acids together in our protein is going to determine its ultimate shape. So we have some things that are going to be more straight chains. We're going to have some things that are more branched. We're going to have some things that are a little bit more globular in shape. So there are many, many different shapes. And that shape is then going to determine its function. We have some things that are very simplistic. We are going to have some things that are much more complex in their structure. And we'll talk later about the exact things that protein does in our body. But we'll see that there are some things like a muscle fiber versus an enzyme. And both of those are made from protein, but have very different structures and functions in the body. So protein synthesis and organization, again, we're linking lots and lots of proteins together by chemical bonds. So we linked amino acids together, and now we're also going to be linking whole proteins together perhaps, and they're all that attraction of those acid and those amino groups that are going to come and pull that together and initiate that formation of the peptide bond. So bonds are going to form between amino acids, the amino group and the helps if I can read. Bonds are formed between the amino group of one amino acid and the acid group of another, which is what I had mentioned earlier. So those are going to be peptides, and they're going to be dipeptides, which is 2 groups. Tripeptides are 3 groups. Oligopeptides is 4 to 9 groups, and polypeptides are 10 or more groups. So again, you can see we might have something that's fairly simplistic to something that is much more complex. Most of our proteins are going to be those polypeptides because their structure is going to be much more complex, so that 50 to 2,000 groups of amino acids. In order to make this happen, we need to really think about how that protein synthesis works. So we'll see that in order to create a protein, it's all about DNA, deoxynucleic acid. And so that DNA contains the information necessary to produce proteins. So everyone's DNA and every cell's DNA and every part of your body that requires proteins DNA is going to have to copy. So as we are doing that copying process, the transcription or copying of a segment of DNA results in mRNA, which is a copy of the information in DNA needed to make that protein. So think about copying a message or a report or something like that. The mRNA leaves the nucleus and goes out into the ribosome of your cell. And from there, amino acids are carried to the ribosome by tRNAs containing the code that matches one on the mRNA. So basically, it's like, okay, so we need a leucine here, and we need an isoleucine here, and we need a valine here. And so your body pulls those in and starts to plug them in. In the process of that translation, the information contained in mRNA is used to determine the number, types, and arrangements of amino acids in the protein. So hopefully we are able to copy correctly. Unfortunately, we know that sometimes that copying doesn't happen absolutely, and there are mistakes that are made. And when those mistakes are made, we are going to end up with a cell that doesn't function as well, which sometimes leads to things like the cancer process, the aging process, and the more of those cells that don't function, the more problems our body has. So our body tries really hard to make sure that it copies those correctly so that we end up with the correct new protein. So that concludes our lecture on protein structure.